Hello and welcome to today's webinar session on microfluidics technology in viral detection in infectious disease research. In a short period of time, a novel coronavirus, SARS-CoV-2, has impacted our lives, society, and healthcare infrastructure. In today's webinar, we will provide examples of how Fluidigm products are supporting global laboratory efforts in response to COVID-19 to address critical needs for testing and clinical research by delivering the following: versatile automated microfluidic systems that support multiple biomarker types, for example, DNA, RNA, and protein. Scalable throughput that enables the testing of more than 6,000 samples per day with multiple targets of interest per sample. Flexible panel designs that enables users to easily add, remove, or change assays without changing technologies. And cost-efficient workflows that minimize reagent consumption using microfluidics technology. During this webinar, we will explore how Fluidigm multi-omic solutions help enable interrogation of health and disease in comprehensive ways. Whether you're seeking to gain new insights into SARS-CoV-2 viral detection or just infectious disease research, or to accelerate therapeutic development, we can help you reach your goal. At the end of this 35-minute presentation, we'll have a live Q&A session. Feel free to type your questions in the question box at any time during this presentation. Part 1 of this webinar features detection and characterization of viral pathogens with microfluidics-based genomic analysis presented by Bill Hunt. Part 2 of this webinar features detection of human and viral full-length RNA using a nanoscale microfluidic platform presented by Steve Kain. At this time, I'd like to introduce Bill Hunt, Director in Microfluidics Product Management at Fluidigm. Bill, take it away.
Today's COVID-19 challenges are broad and wide-sweeping as they continue to tax healthcare systems around the world. We've seen significant impacts on first responders, hospital infrastructure, as well as the access and supply of critical resources, including personal protective equipment, as well as tools and tests for detection. Critical needs are many and include rapid, high-throughput genetic and epigenetic testing, immune and serological monitoring, as well as antibody response and surrogate marker identification to evaluate vaccine efficacy. Taking a snapshot of the virus, there are a lot of great resources available today. I've been using the WHO website as well as the Johns Hopkins website to give updated tracking information on a daily basis. Just looking at the numbers from April 1st, you can see there's a large number, a tremendous number of affected individuals worldwide, as well as a sizable number of deaths, which continues to grow on a daily basis. And the countries that have been most affected based on the numbers are US, Italy, China, Spain, and Germany. So this is truly a global pandemic. You can also see Iran has been affected in the Middle East. South Korea has been affected, but has done a tremendous job at countering the virus. So it really does prove that having an early response, having an orchestrated response is an effective way to deal with pandemics such as this. In light of this disease has only been on the world stage for about 3 months now, as it was first reported in Wuhan, China in December of 2019. Of course, it's since spread globally. Also wanted to touch points on nomenclature. I'll be using the term SARS-CoV-2, which seems to be the favored term used these days, but it's also been referred to as novel coronavirus 2019, and the disease itself is referred to as COVID-19. There have been a lot of comparisons with SARS-2 or COVID-19 to flu. And I'm using this graphic, which is a wonderful resource that was put together by Vox, which kind of encapsulates all the details in one simple graphic. So if you look at the reproductive number, or the RO number, this reference is the number of individuals that are infected by a person who's infected themselves. So if you have a RO value of 1, one person is infecting one other person. If you have an RO value of 2 or 2.5, as you're seeing for COVID-19, one person is infecting 2 to 2.5 individuals. And I've seen references that this can be much higher depending on the individual who's infected and the transmission rate of that individual. There's also the incubation time. Flu has a relatively short incubation time between being infected and showing symptoms of 1 to 4 days, whereas COVID-19 has a rather wide window of infection to a person becoming symptomatic. It can be anywhere from, on average, 5 days, but there are reports of individuals becoming symptomatic on 14 days. The hospital rate is much higher for COVID-19, which is part of the issue. It's happening so fast that healthcare systems globally are just not able to respond to it in the time frame needed. And the case fatality rate is much higher for COVID-19. Overall, it's relatively low for flu. It's proving to be upwards of 3.4. I've seen numbers over 4 now for COVID-19, depending on the country. The technical requirements for battling a pathogen such as COVID-19 include the ability to scale sample throughput. We're getting a lot of requests for sample testing around the world, and the capacity is just not able to keep up with demand. So being able to support high volumes of samples for viral detection and biomarker research is critical. Maximizing results from precious samples is also a factor for consideration in certain technologies where the sample may not be sufficient volume. So being able to get as much out of a single sample, as much testing as you can out of a single sample is important. And then finally, cost efficiency, being able to deliver high quality data and services with limited available personnel and budgets. So Fluidigm is supporting global laboratory efforts in response to COVID-19 in a number of ways. On our proteomics side, we have leadership in highly multiplex cytometry and imaging products, which maximize results from precious samples with proven CyTOF technology. On the genomic side, we have versatile, scalable microfluidic platforms, which are systems that support multiple biomarker types, including DNA, RNA, and protein, in applications with a range of different throughputs. Both technologies offer flexible panel design, so you can easily add new biomarkers to existing panels without changing technologies. And we offer simplified workflows and automated platforms for improving productivity of the lab and driving return on investment. So focusing on the Fluidigm portfolio of products, I'm showing in this figure the CyTOF technology products on the top part of the figure and the microfluidics technology on the lower part of the figure. As I mentioned in the introduction to today's presentation, I'll be focusing on the genomics and gene expression component of our portfolio. Moving into SARS-2, which I easily truncated, it's easier to say SARS-2 than SARS-CoV-2. I wanted to touch base on the phylogenetic and genomic details of the virus itself. So in terms of its classification, it's together with other pathogenic coronaviruses, such as the original severe acute respiratory syndrome virus, as well as the Middle Eastern respiratory syndrome virus. All three are from the genus beta coronavirus. And the genome is positive sense, single-stranded, polyadenylated RNA of about 30,000 kilobase or 30,000 bases. And the reference genome is available in GeneBank, and I have a hyperlink shown on the slide. The genome consists of, it supports 4 major structural proteins. There's the spike glycoprotein, which is a key target since that is the protein that enables the virus to access and enter the host cell. So it's a target for several therapies as well as vaccines. But there are additional structural proteins such as the envelope, membrane, and nucleocapsid. The figure at the bottom just shows the 3 pathogenic coronaviruses and shows how closely related they are genetically, which is not to be unexpected considering they all are from the same viral genus. So Fluidigm products can be used to detect and study COVID-19 in a number of ways, starting with screening and detection. This I've broken out into two structures or 2 elements. There's the qualitative element, which is seeing if the target is present or absent, but you could also use it to say, is a genotype present or absent? That can progress into the quantitative type of a detection, more often referred to as a viral load, where you're looking for the amount of virus present in an individual at any given time. And these methods often rely on PCR-based assays to support them. Shifting over to the other element of the spectrum, you've got clinical research, and there's a whole host of open questions regarding the virus, regarding the disease, and how individuals are reacting to the virus. So that's in active areas of research by a lot of individuals. And that includes viral evolution, looking at the phylogenetic analysis, mechanisms of action such as population stratification, who's responding to the virus in a more negative way than other individuals, transmission of the virus from person to person, virulence, host resistance, and virus susceptibility to various compounds in development by the pharmaceutical industry. And this can involve a varied set of tools, which includes next generation sequencing as well as PCR. The Fluidigm portfolio, the genomics portfolio for Fluidigm has several components, which I wanted to touch on here. Starting with our instrument systems, you'll see it in subsequent slides. We refer to the Juno system, which is a system that supports NGS library prep. We also have a Biomarker HD system, which is our universal PCR system. It supports endpoint PCR, quantitative PCR, digital PCR, which can be used in a variety of applications. We also have our integrated fluidic circuit technology, or IFCs, which may also be referred to as chips. And these are devices that support tens to thousands of nanoscale reactions per sample. We also provide reagents that are used with our chips, and these are fully kitted, and they offer high performance materials from a single supplier. And then finally, we have, for custom needs, we have our D3 assay design services. So if you're looking for designing assays for certain targets, if you need help designing assays, this would be a service we make available to you to help you facilitate the design and implementation and development of the assays. Focusing a little bit more on the instrument components themselves, I mentioned the Juno system is our system for library preparation for targeted sequencing, but we refer to it more as a preparatory system because it also is a universal controller. It is used to process all formats of our microfluidics, and they can support a variety of PCR applications. So if you take our integrated fluidic circuit, use Juno to process that IFC, you can then transfer it to the Biomarker HD for PCR-based analyses, such as gene expression, genotyping, copy number analysis, and digital PCR, or you can have it go to Illumina next-generation sequencing for targeted library prep, and it also supports an RNASeq application. Giving a little bit more background on the integrated fluidic circuit itself, it's an integral component of our portfolio, and it helps us automate molecular workflows at very small volumes. And so kind of customizing this for how this can help with COVID-19 work, it is useful to have an adaptive system in place. So as the virus adapts, as needs adapt in the lab, the IFC can adapt as well. So it does enable automated high throughput workflows with rapid turnaround time. It offers scalability. So based on the different types of IFCs we have, you can support a small number of samples, a larger number of samples, a smaller number of assays, a larger number of assays. So in this case, I'm showing a 192/24 dynamic array IFC. And the nomenclature here represents the 192 on the left is the number of samples. The 24 on the right represents the number of assays. So each of the samples on the IFC is interrogated by each of the assays on the IFC using the microfluidic matrix in the center of the IFC. It's designed to be dynamic, meaning that you can add, remove, or replace assays on demand because the assays are not prefixed or pre-spotted. So as the needs change of the application, you can use open inlets to supplement. You can easily swap out one assay for another as needs change. So it's a very nice design for adaptability. And it also is designed to support different applications. So you can analyze multiple pathogenic targets simultaneously to help identify the source of respiratory diseases that share common symptoms. The IFC is designed to also help with cost savings and operational efficiency. And the best way to compare it is to use the benchmark for what's commonly used in labs today, or historically has been used in labs, which is the microtiter plate, whether it's the 96-well microtiter plate or the 384. And if you started doing a comparison, you'll see from a data point perspective, one of these IFCs, and here I'm showing a 96/96 IFC, one IFC supports the equivalent of 96 96-well plates. That means you get the same amount of data from one IFC as you'd get from 96 96-well plates, or you get the same amount of data from 24 384-well plates. And the other benefit is because we're operating on a much smaller scale, we're operating at the nanoliter scale, you use far less reagent. So it's the efficiency of the throughput, the amount of data you get, coupled with the small amount of reagent needed to process a sample, helps you get a very efficient workflow and a very cost-effective workflow. The D3 assay design portal, I wanted to come back to this because I touched on it earlier, and I wanted to share a little bit more background about what we've done using D3 as it pertains to COVID-19 to date, and what we're hoping to do as we continue to engage with customers. So we can design assays that are specific for coronavirus using publicly available genomic reference sequences and primer designs. For example, we can use the NCBI reference sequence shown here And we have also designed assays that are specific to the spike, the S, M, N, and E gene sequences of coronavirus. And we've been able to design assays specific for a panel of respiratory pathogens using the example shown here. And the way to access the panel, or a way to access the portal is provided at the bottom of the slide. There are multiple ways to get assistance. You can email the assay design group using the email shown on the slide, or you can contact your local sales representative or field application scientist for more information. Moving into the biomarker products specifically, how these products can be used. The benefits of qPCR using biomarker are listed on the slide, starting with rapid detection. Our current workflows allow results in less than 4 hours, and they also support high volume screening. So you can screen up to thousands of samples per day with flexible batch sizes based on the IFC format you use. And the IFC format is shown on the right side of the slide. It also supports the adaptable assay content that I referred to earlier. By having different designs, you can either run up to 24, 48, or 96 assays per sample, and it's compatible with different technologies. So you can we can design assays that are common to publicly available products such as those from BioFire, Luminex, and Thermo Fisher, which cover approximately 20 to 30 targets per panel. We can easily add new assay targets as new strains emerge. This is one of our benefits of the IFC, as I mentioned earlier. And we can target pathogenic strains of coronavirus themselves, since there's four strains that generate mild disease in individuals, plus the 3 pathogenic strains. Or we can target a broader array of respiratory pathogens that share common symptoms, so it's easier to identify the causative agent in a given individual. Switching to our digital PCR products, they share many benefits, including sensitive and absolute quantitation. Because it's digital PCR, we're able to have sensitive viral detection using the digital PCR IFCs, which are shown here on the table on the right. And they support up to 12 or 48 samples per run and up to 770 digital partitions per sample. The workflow is fairly quick. You get results in less than 3 hours, and it provides users with additional flexibility. You can prepare 1 or more sample mixes using gene-specific assays of interest, which allows you to process all your samples with the same assay, or as needs arise, you can process samples with different assays. Biomarker is associated with a wide number of publications, quite a good portfolio over the years. Today, I want to focus on those that are specific to pathogen detection, which is relative in the case of COVID-19. So selecting a few to present today, I selected this one, which the title is Disclosing Respiratory Co-Infections, a broad range panel assay for avian respiratory pathogens on a nanofluidic PCR platform. And this research was conducted by a group in France in 2018, which had an objective of demonstrating the ability to come up with a high throughput screening protocol to detect respiratory pathogens in poultry. They used the Biomarker HD system with the IFC controller HX, which is a precursor to the Juno system. And they also used the 9696 dynamic array IFC. And they concluded that this workflow allowed them to develop that high throughput workflow that they were looking for a multiplex panel targeting several respiratory pathogens that included viral, bacterial, and fungal targets. Switching to the second publication, this is also a group from France that published in 2016. They were looking at human pathogens, so similar goal as the previous authors, but this group was really looking at human pathogens, and specifically human pathogenic viruses. And they wanted to do a quick, rapid, high throughput method, and they chose the Biomarker HD system to do so. So their goal was to select a panel of 19 human pathogenic viruses and controls. They ran this on the Biomarker HD with 2 of our digital or dynamic array, digital array IFCs, the 48.770 digital array IFC and the 4848 dynamic array IFC. And they concluded that the combination of these 2 products allowed them for high throughput sample screening during an outbreak investigation and surveillance into human pathogenic viruses. The remaining slide is just 3 additional publications that I thought were worth consideration, including in today's presentation. I won't discuss them in detail here, but I will point out the last presentation at the bottom of the slide is a publication from Turkey in 2014, where they were looking at different plant compounds and how they affected the replication of coronaviruses. Not this particular coronavirus for COVID-19, but earlier coronaviruses. So this may have some applicability in modern times. So in terms of engagement with customers, we're actively trying to work with our customers who are very much interested in coming up with solutions in response to COVID-19. And one group in particular I'm highlighting today per a press release we had in mid-March. And this is a group at Mount Sinai, and they are leading a consortium to develop real-time PCR tests for coronavirus using the Biomarker HD with our IFCs and Roche Universal Probe Library products. And their goal is to produce a multiplex epigenetic and RNA system to support high throughput screening and detection both during and post incubation. So their goal is really to see how early they can detect virus in individuals after infection. It's based on the earlier slide I presented from Vox. The incubation time up to the presentation of symptoms can be quite long, up to 14 days, perhaps longer in other individuals. So it's important to be able to detect virus as quickly after infection as possible to ensure that they can be triaged appropriately. And the consortium is planning to submit this to the US FDA for emergency use authorization. Moving into RNAseq, in addition to our PCR products on Biomark, as I mentioned, we have our NGS portfolio as well, which includes the Advanta RNAseq NGS library products, which can be used for SARS-2 analysis. It supports walkaway automation, substantially reducing pipetting steps and operator interventions by putting a lot of the content, a lot of the workflow onto a new IFC we've developed, which I'll show in a moment. There are superior cost savings, which allow you to maximize laboratory budget and minimize consumption of reagents and consumables based on the use of our microfluidics. And the chemistry is robust, allowing you to confidently generate high-quality, full-length stranded RNA sequencing libraries from a variety of organisms, as long as they are all polyadenylated, which includes the coronavirus. So I mentioned the new IFC. This is our newest IFC to the portfolio. It's called 48.Atlas, and it's designed for solid phase capture in multi-step reactions. I won't discuss each of the steps shown below the IFC, but as you can see, a lot of the workflow steps that would normally be part perhaps of a manual workflow, we've been able to integrate in an automated fashion on the device itself. So it allows you to, once you've loaded the reagents and you've loaded your samples and assays onto the IFC, transfer this to the Juno system, it becomes a walk-away workflow. In showing how this might be used in context, this is a publication that came out in 2019 for the analysis of COVID-19 host gene expression changes from the same sample. So it's an example of how this might be used. So you've got gene expression analysis being critical to understanding the host immune response to coronavirus. And you've got the 3 pathogenic coronaviruses, which all have positive sense single-stranded RNA genomes with a polyadenylated tail. And because of this, this makes it suitable for use with the Advanta RNAseq products in our Advanta RNAseq workflow. And by using this, some examples of what you could do as part of the COVID-19 research include hypothesis-free measurement of phenotypic changes in cells or tissues in response to viral infection. You can also characterize immune cell gene expression in patients at different time points after viral infection. You can characterize COVID-19 sequence variants and the impact on host gene expression. And you can identify immune cell expression signatures that correlate with clinical outcome to guide treatment options. So kind of to reiterate some of the earlier points, we are actively engaged with global responses to COVID-19, and we feel that our products can be helpful in that effort. We are a leader in highly multiplex cytometry and imaging technology that involves CyTOF. We offer versatile and scalable microfluidic platforms for a variety of applications. We offer flexible panel designs so that you can add, remove, replace assays as needed. And we offer simplified workflows with automated platforms that provide operational efficiency and cost efficiency. So in closing, I'd like to reiterate global efforts and global request for everyone to shelter in place. But while we're sheltering in place, hopefully everyone remains in touch through virtual means such as our website and other social media. I just wanted to make you aware of some things we've put on our website to help communicate activities we're undertaking to address COVID-19. You'll see that there's the COVID-19 update page on the right, to which we will regularly update content so that you can keep appraised of our activities and what we're doing to help your laboratory and other laboratories stay ahead of this virus. Thank you.
Thank you, Bill. For the second part of the webinar, I'd like to introduce Steve Kain, who is a director in Genomics Product Management at Fluidigm. Steve will walk us through our virtual poster, Detection of Human and Viral Full-Length RNA Using a Nanoscale Microfluidic Platform. Whenever you're ready, Steve.
So to begin with, I have just a brief outline of what I'll be covering. Introduction, a methods and materials section to describe the analytical study that I'll be showing. I have a few different views on the workflow for our RNAseq library construction method. And then I'll cover the results of our analytical validation study, which assess the technical reproducibility of the platform against different operators, and also a comparison of gene level detection of libraries produced with the Advanced RNAseq system, as well as another commercially available RNAseq methodology. And then I'll have some brief conclusions at the end. So to begin with, the Advanced RNAseq NGS Library Prep Kit is a consumable kit that operates on a microfluidic platform called the Juno instrument. So using the consumables and the Juno instrument together, you're able to produce 48 independent RNAseq libraries from 48 RNA samples using a one-click script on the instrument. This is a poly-A capture methodology. So for human RNA, you're looking at the mRNA population, but because the coronavirus genome is also an RNA that's polyadenylated, you're able to look at both viral and human host sequences in the same RNA sequencing experiment. Together with the instrumentation and the consumables, we've also developed a unique integrated fluidics circuit, or IFC, called the 48-dot atlas. I'll show you on the workflow diagram how this is utilized together with the Juno to produce random prime stranded RNA-seq libraries that, again, are polyadenylated using an input of as low as 10 nanograms total RNA. And this is compatible with any polyadenylated RNA, be it from eukaryotic organisms or viral species. The entire workflow can be completed in about 12.5 hours with 2 hours of hands-on time. So in terms of materials and methods, in the validation study, we looked at just over 900 different total RNA samples. These are from the universal human reference RNA or human adult brain RNA. In order to look at the reproducibility of the system, we looked at the production of libraries from 3 different independent operators running the workflow, using 3 different lots of our integrated microfluidic chip, 3 different reagent lots, and 6 different Juno instruments. The libraries that are produced are compatible with the Illumina platform, and so in this study we use both the MiSeq and the NextSeq system. So this diagram shows you the overall workflow and how the instrument, the IFC, and the reagents are integrated. So in step number 1, the initial step by the operator would be to load the total RNA onto the IFC. This is typically done at the end of day 1 of the protocol. It takes about 60 minutes to load the IFC and then prepare it for loading onto the Juno instrument in step number 2. The Juno then runs for about 9 hours to complete the automation script. Most people do that in an overnight run, and then in step 3, collect the libraries at the beginning of day 2. After the libraries are collected, then off the IFC, there's a cleanup procedure. The adapter PCR is run to amplify the library, QC, and normalization. And then the final output then is 48 RNA-seq libraries that are ready for sequencing. So you can see in the red circle, that's just indicating where the IFC is loaded on the Juno instrument prior to the overnight run. Looking at a bit more detail then on the steps that are automated within the IFC, or the microfluidic circuit, if you will. Total RNA is loaded again at the first step. The poly-A fraction is collected using an oligo-DT column that's on the IFC. In step number 2, the RNA is fragmented. It's then reverse transcribed and template switched to add the sequences necessary for the Illumina adapter formation. Sample barcode PCR then amplifies the library, and as I showed on the prior slide, the libraries are then harvested, QC'd, amplified, and prepared for sequencing. So noting the volumes indicated for each step, you'll see that this truly is a nanoscale volume, nanoliter volumes taking place in each reaction. So what we've done here is miniaturized many of the steps used in RNA-seq library production. This allows us to significantly reduce the cost of reagents required to run this workflow. And then the last workflow slide is just a comparison between the Advanta RNA-seq system shown on the left and another commercially available system from Illumina called the TruSeq Stranded mRNA Library Prep Kit. So this is a manual workflow. Draw your attention to the rectangle on the left, which shows the steps that are automated on the ISC. So these are fully automated and complete walkaway automation. So the overall time to complete the workflows are similar, but where the difference is the hands-on time. So for the Advanta RNA-seq system, it's about 2 hours. In contrast, the TruSeq workflow requires 6 hours and 45 minutes of hands-on time. This data figure is an overall summary of the sequencing performance results and mapping statistics that were generated in the validation testing. So we use 2 different inputs of total RNA, either 10 nanograms or 100 nanograms of the universal human reference RNA or the adult human brain RNA. And I will draw your attention to the rightmost column, which averages all of these results. So you can see that the percentage of reads mapped to the genome is quite good, 84% on average. Percentage of reads mapped to the transcriptome, about 73%. This is a poly-A capture methodology once again, so we're able to exclude ribosomal RNA sequences. And so they're quite low, on the order of 5.4% overall average, down to as low as 3%. We also looked at the correlation between results using either 10 or 100 nanograms in the next to the last row. And you can see that those correlation coefficients are quite high, 0.981. So that would indicate that you get comparable results over that dynamic range of inputs between 10 and 100 nanograms. And then lastly, this is a stranded protocol producing stranded RNA-seq libraries, and we're getting correct strandedness that approaches 100%, 98.3%. This next figure shows the operator variability. So we wanted to look at the concordance of results between 2 different operators. So in this experiment, either 10 or 100 nanograms of each RNA type were run. And we looked at the gene level Pearson's correlations between these 2 results. And you can see there's a very high level of concordance between each operator, indicating good reproducibility based on 2 different operators running the process. In this figure, we looked at the level of gene detection between the Advanta RNA-seq system shown in red and the TruSeq systems shown in gray at 2 different inputs, again, either 10 or 100 nanograms. And we're essentially seeing comparable level of detection for genes with either workflow. Not shown in the poster is the similar experiment where we looked at the number of detected transcripts. Those were also very similar with each workflow. So you are getting comparable sequencing performance results, but with the Advanta RNA-seq system, you're able to obtain those results with significantly lower hands-on time and reduce cost per sample for reagents based on the use of microfluidics. This slide shows you the overall portfolio of applications that can be run on the Juno system. So on the left, in terms of library preparation, in addition to the Advanta RNA-seq NGS library prep kit that I've discussed, we also have a number of standard panels for targeted sequencing, and also the D3 assay design portal allows people to make custom panels for targeted sequencing. All of those libraries have adapter sequences that are compatible with the Illumina sequencing platforms. On the right side of the figure shows that the Juno can be utilized together with another instrument from Fluidigm called the Biomarker HD, and this would be for PCR-based applications such as gene expression or genotyping. And then we also have a similar custom design capability for both gene expression and genotyping panels that are designed by customers. So in conclusion then, with respect to the RNA-seq results that I've shown, using the Advanta RNA-seq NGS library prep kit, you can obtain excellent genome mapping rates, transcript detection, high level of correct strandedness with low percentage of reads going to ribosomal RNA sequences. The technical replicates are quite good, so we showed results with greater than 98% concordance. The platform study showed performance that was as good or better than the TruSeq kit for gene level detection across the dynamic range of 10 to 100 nanograms input and across a large range of detection for different transcripts at different expression levels. The Advanta RNA-seq NGS library prep kit provides an automated solution, so you're minimizing the use of manual pipetting steps with a consequent reduction in hands-on time and truly increased walk-away automation. In addition to the results that I've shown for human reference RNAs, we are in the process of doing studies related to COVID-19 research to look for the ability to detect viral sequences that are polyadenylated against a background of host mRNA in the same sequencing run.
Thank you, Steve.